Hello students, welcome back to the channel. In this video, we are going to study cellular component of second line of defense. Before this, understand two type of immune systems are there. One is innate, which is non-specific. One is adaptive, which is specific. First line of defense will include skin, mucous membrane, and certain chemicals. They are trying to prevent it. But if suppose pathogen gains an entry, then the second line of defense will start. And second line of defense will have two type of component. One is cellular component, another is non-cellular component. So my focus today, we will be discussing about cellular component, which plays role in the second line of defense. Now see, important cellular component are phagocytes. They are doing phagocytosis. Example like neutrophils, macrophage, and dendritic cell. Then is natural killer cell, which are non-phagocytic cell, but they will kill abnormal and virus infected cell. Then is mast cell, which will release histamine to bring the response of the inflammation. Then is eosinophil, which is releasing these enzymes to destroy the pathogen. Is it clear? So these are the cellular components which play very important role in the second line of defense. Now let us understand one by one. See here phagocytosis, which is done by neutrophil and it is done by macrophage. So what is the first step? First step is that pathogens will be coded by the complement protein, which is non-cellular part, okay? The pathogen will be covered by complement protein so that now pathogen becomes easily identified by the phagocytes. So phagocytes are able to easily identify the pathogen. Is it clear? So first step is adherence. It starts when the phagocytes will attach to the pathogen. And how will they identify this is pathogen? Complement protein will do the process that is known as opsonization. Means they will coat it on the surface of the microbes and so that phagocytes can identify it. Then second step is ingestion. Means now they will give rise to pseudopodia so that the pathogen is engulfed inside. Is it clear? And after this ingestion, they are making the food vacuole known as phagosome. Now this phagosome combined with lysosome. Lysosome you have already studied in the cell. It is a vesicle rich in the enzyme. After that, digestion will be done. Okay, all the components will be broken down. Biomolecules, carbohydrate, protein, lipids, they will be degraded down. Is it clear? Then the material which is not able to degrade, that is known as residual body. So 4 steps of phagocytosis. First is adherence, which is helped by the complement protein. Then is eating, that is ingestion, which is a type of endocytosis. Then is digestion, which is done with the help of lysosome. And last is killing to break down all the components into the simple form. Now this is natural killer cell. This natural killer cell will not do phagocytosis. What they will do, suppose this natural killer cell will bind to the infected cell. Then they will release some chemical. So natural killer cell will release some substance that is granules. Now these granules will release perforin. It is a protein which create a pore in the target cell membrane. Okay, so that water will enter, extracellular fluid will enter and the cell will go for bursting. Some granules will release granzyme. So granules will release two things, either perforin or either granzyme. Granzyme is a protein digesting enzyme which enter the target cell through this perforin channels and induce the target cell to undergo apoptosis, means programmed cell death. Is it clear? This is the way how innate cellular defense work. So in this video, I try to explain you only cellular defense, which is playing role as a second line of defense. So I hope so this video might be helpful for you. Thanks for watching. And if you like, please subscribe to my channel. That will be motivating for me. Thanks for watching.